Hi, I am Manuela Bonanno from Columbia University, and today I'm very delighted to meet Katherine Morel. She's the winner of the Radiation Research Journal Editor Awards for this year, 2018. And she published, actually in 2017, right? Your article entitled "Parthenolide Selectively Sensitized Prostate Tumor Tissue to Radiotherapy with while protecting healthy tissues in vivo." And let's start by introducing yourself. So, where did you do these studies, and where you are now?
So the work that I did for this paper was for my PhD research, which I did at Flinders University in South Australia. So in the lab of Professor Pam Sykes, who's got a long track history doing low-dose radiation research and is quite well known within the society.
And where are you now? Are you still in Pam Sykes' laboratory?
No, so I finished up my PhD research last year and have now moved over here to the US. So I'm at the Dana-Farber Cancer Institute in Boston.
So when you were talking about Pam Sykes, she always does very intriguing research.
Yeah.
And this particular topic that gave you this wonderful award, congratulations by the way, I think it's very nice because it sounds like you found the Holy Grail. So you have this fantastic drug, molecule, that can selectively kill tumor cells, but be nice and spare normal cells. And so tell me if this is true, you found the Holy Grail, and how radiation fits into it.
Well, I don't know if it's the Holy Grail, but it's certainly, we're very excited about it because it seems to be doing a lot of all the right things. So really interestingly, we came across this compound because of Pam's low-dose radiation research, and when we were looking through the literature, what we found was that this compound had very similar properties to low-dose radiation, so where we see low-dose radiation protecting from, say, higher doses of radiation.
So the sort of like anti-inflammatory, or?
Yeah. So we were noticing just from reports that it had, it seemed to be protective of healthy tissues. So that really interested us. But unlike what we've seen with low-dose radiation, it also had radiosensitizing properties in tumour, which was what we were really interested in looking at. So in a nutshell, the work that we did, we essentially treated mice that had so autochthonous prostate cancer model of mice, treated them with high doses of radiation while giving them this drug Parthenolide. And we noticed that the tumour tissue was indeed radiosensitized quite significantly, and we actually protected the healthy tissues within the one animal, which was really, really cool.
So you did administer the drug beforehand to the mice?
Yes.
And then?
Yeah, so we gave the mice a week of priming with this drug just to get it into the body. And then irradiated them, for this paper we irradiated them with a single high dose of 6-Gray and looked a few hours later to see the effects of the radiation-induced apoptosis in these mice.
So what did you find?
Well, what we found was that, as I said, in the healthy tissue, so we looked in spleen and colorectal tissue, we found a significant reduction in the amount of radiation-induced apoptosis. Whereas in our tumour tissue, in the mice, we upregulated the radiation-induced apoptosis. And what was really interesting, when we looked at these mice that had a range of tumour development, so some had very low-grade tumours, some had high-grade tumours, we saw the most, the most amount of radiation-induced apoptosis in the mice that had the highest grade tumours. So the drug was really able to select those high-grade tumours and affect them much more so than the lower-grade tumours, which, when we had a look at them, seemed to be to do with the underlying level of oxidative stress in the tumour tissues. So the higher the baseline oxidative stress in these tissues, the more they were affected by the drug.
So you used a word that caught my attention.
Mm-hmm.
You said you primed the mice with the drug. Mm-hmm. And this reminds me of a nice classical or fundamental radiobiology mm-hmm. protocol of adaptive response.
Exactly.
So did you explore or did you compare like giving a small amount of radiation before the 6 gray and see if you would got?
We did. So one of the things that interested us, as I said, we noticed that the low-dose adaptive response seemed to be similar to the protective effects that we were seeing with Parthenolide. But some work that's come out in recent years, I think some work by Dave Gradina showed that while low-dose adaptive responses can protect healthy tissue, they can also protect tumour tissue, which is incredibly problematic for patients who these days, particularly for prostate cancer, receive image-guided radiotherapy. So you end up with a low priming dose before your actual therapeutic dose. So we wanted to see if we could combine Parthenolide with a low dose of radiation, whether we would have additive protection and stop any protection of the tumour. So we did some initial studies, and at least the ones that we presented in the paper, and we didn't see a very, very big additive protection in our healthy tissues, but certainly we weren't seeing any protection of our tumour. So it's an aspect that we'd really like to explore in a little bit more detail, and certainly looking much more at that sort of clinical protocol where you do have daily fractions with daily low imaging doses, just to see whether there is some interaction there.
So this is another $1 million question. What's the mechanisms by which the drug can selectively kill tumor cells but protect in a way normal cells?
That is the $1 million question and something that we're still working through. But we think the fact that this drug, it works through a couple of different mechanisms. It's most well known as an anti-inflammatory drug through inhibition of NF-kappa B. And what we see in cancer cells is that it's able to inhibit double-strand repair in tumour cells through that NF-kappa B inhibition pathway. So not just us, but a number of other people are still trying to work our way through how that works. But the flip side of it is that the drug also induces reactive oxygen species. So what we think is happening, at least in our healthy tissues, is that when we deliver Parthenolide, it induces reactive oxygen species within the cells, but just enough to induce an adaptive response. So what we see is that when we look at some of the redox pathways, there's KEAP1, which gets oxidized and releases NRF2 to translocate into the nucleus, and that upregulates antioxidant pathways. So in a priming sense, that primes the cells to be in a state of already wanting to protect itself from a subsequent radiation exposure. Whereas cancer cells, because they have that high oxidative stress, they're pushed beyond the point that they can adapt. So the Parthenolide kind of pushes them over the top and really pushes them towards cell death. And that, combined with the fact that the NF-kappa B inhibition is stopping that double-strand break repair and non-homologous end joining, it's sort of like a perfect storm.
Interesting working hypothesis. Did you have the time to dissect and prove that that's what it's happening.
No. Unfortunately, I learned early on in my PhD that there's just only so much you can do as a graduate student. So we did a lot of the animal work, and we've got tissues that we're working through, really trying to work through the mechanism with, and we did some very initial studies. But luckily, there's a lot of people that have kind of taken this a little bit further. So it's the wonderful thing about science, that it's not just on us to work it out. It's a collaborative effort. So there's a lot of people all over the world really trying to work out this mechanism. So it's everyone taking their own little approach to bring it all together.
So you were mentioning that you defended your thesis last year, and now you moved to the US. So what kind of work you're doing there? I think you're working with one of the collaborators, one of the co-authors.
Yes, of my paper. Yeah, so Professor Christopher Sweeney, who I've gone over to Dana-Farber to work with, he's a wonderful medical oncologist over there, and he's pushed a lot of the work that's been done with Parthenolide. So he's got a real vested interest in seeing it move to patients. And so that's a lot of what I'm doing at Dana-Farber, and it's a fantastic institute really to see excellent translational medicine. So that's what he and I are really interested in seeing, is trying to move Parthenolide into the clinic in a number of different avenues. So particularly at the moment, what I'm really focused on is patients who have really aggressive metastatic prostate cancer. So seeing if we can use the drug to really benefit them, because they really have a pretty terrible prognosis.
Right. So I don't know, we might cut this, but the metastatic, it really reminds me of a mechanism of action that you might want, again, Parthenolide to do, like an abscopal action. Can we talk about that, or it's too far off, you think?
Oh, no, it's not too far off. Pam and I talked about looking at sort of abscopal effects and bystander effects in our animals. And we actually did some studies more recently, so just before I left Australia, we did a study where we did, rather than irradiating the whole animal, we pre-treated them with DMAT or Parthenolide and just irradiated the prostate area. So we collected tissues from all over the animal, so we could theoretically look exactly at that kind of abscopal, kind of off-target effects, but sadly haven't had the opportunity to, but we would love to.
I wanted to ask you, so you have been awarded this very, very important award from the Radiation Research Society, and you have been a member of the society for how long?
Almost 5 years. So I joined as an SIT when I started my PhD back in 2013. So I've been involved in the society for quite a number of years now, and I've really loved it. And I think, particularly as a trainee, as a graduate student, it's just been really excellent. And being able to come to these meetings and have the SIT workshops and meet other people in my same sort of at the same point in their careers has been really fantastic.
That's great. What else? Is anything else that I didn't ask you that you might want to touch, or I don't know. Somebody in your group, if you want to thank somebody, or
I guess Pam would be the biggest one. She's been, her work with radiation has, and just her career trajectory has really inspired me, and she's been a wonderful mentor throughout my graduate studies, and really continuing on now. I'm a postdoc and overseas, and she's just been a wonderful support, and has always really pushed me to put myself out there and take every challenge I can, and I think I wouldn't be where I am today without her. So she's been wonderful.
And then another question, and again, I'm not so sure if we can blend it in, but I was curious, so we keep the camera rolling. How was the impact of changing from South Australia to the US?
Oh, well, the most immediate impact was the temperature, which was a big shock. So I moved in the middle of an Australian summer to a Boston winter. So from very, very hot, sort of 40-degree temperatures to snow and blizzard. So that was a little bit of a shock. But it's been really good. I think it's a wonderful... It's been an adjustment moving from Australia to the US, but I think it's such a wonderful place to be working, and the opportunities to work with so many just brilliant minds both at Dana-Farber and Harvard more extensively has really helped that. And I think there are potentially a lot more opportunities here for postdocs and a lot of different avenues that I can sort of take my research than I would have been able to have back in Australia. But yeah, it's been really good.
That's great. And I'm really glad that you are continuing in the field. And I really hope that the Radiation Research Society will, and I'm sure it will, keep on giving you some other opportunities and visibility for your work. And this is the purpose of this interview, for example.
Yeah.
Giving visibility, giving value to the work that you do, and hoping that you might continue in the field contributing. And hopefully I will interview you again for other fantastic papers, and I really wish you good luck for your career.
Oh,
and thank you again for taking the time to talk to me.
You're welcome.